Hello, everybody. My name is Stephen, and I work with a company called SPV Healthcare, and we have collaborated with Shannon ABC over a couple of years now, and with a number of different projects. So a little bit of background, I suppose, to SPV. We are a company based in Clonmel. We have a laboratory down there. We take healthcare products, whether it be cosmetic, medical devices, medicines, or food supplements, and we would take them from concept through to market launch, and maybe all of the different steps in between that. So there could be a concept, or we might need to look at what might be in the formulation, and what are the clinical outcomes, then look at what registration might be needed. If it's like it's a medicine or medical device, does it need to be registered with the authorities? If it's a cosmetic, that's a different registration process. We would look at any intellectual property or so as well, maybe like file some patents or do some prior art research, and then take it through to manufacture as well. And we do these with both products for ourselves, for SPV Healthcare, and also products then for other companies. So with Shannon ABC, what have we done? We have worked with them on a few different projects. Some of them are here. One of them was to grow a specialist microbial organism, a fungus, which was not available from any supplier or so, and it took us quite a bit of while to track that down. And so what we did was we needed to buy some freeze-dried culture, and then to look to incubate that and grow that and keep that then as a supply of that fungus. And that we were able to do with Shannon ABC. We have also done some cell line work and some immune profiling, so looking at different cell lines, looking at the immune responses coming from those. And we have also done some analytical testing and method development. So we have a product that we're looking to analyze some of the active in the product. And it is that particular product that I'm going to talk about. But why did we choose Shannon ABC, and why work with them? There are a couple of reasons. They have quite a broad range of knowledge and specialist knowledge. They work in quite a range of areas, as Tim is saying, and that can be quite beneficial because it can be interdisciplinary work involved. And the range of equipment that's there and the type of equipment is really helpful and quite extensive. And then we can also negotiate and agree into intellectual property terms. And we have done our studies. Most of our studies have been done by the direct funding method, which with the intellectual property as well. So this then the particular project that we talk about now, which is an analytical project problem that we have. We have a gel, which is an aqueous matrix in a polymer matrix, and there are two active ingredients in that matrix which we want to measure their dissolution, how they come out of the gel over a period of time, which was 3 days, and also over a number of pH values, so a number of chemical environments. Because especially in the early stages, the release profile is low, that the limit of detection was low, so that potentially should be aware of. And we had done some method development ourselves, but we hadn't been that successful with it, as we'd like to be. And we got at most maybe with 60% recovery. So of the active that were there, we were only maybe able to recover 60% of them. And so there was a problem with our method as well. So we went with that then to Shannon, and they're with those points, and Shannon ABC was then able to develop a method, tweak the method, and develop it further, so that we actually got 100% recovery. So we knew then that all of the active ingredients that were there, that we were actually able to recover them. Were they coming out of the matrix, or are they not? And we were able to get full recovery. So we were able to consolidate all of the ingredient, the amount of actives that were there. Because the LOD limit of detection was an issue, we used mass spec, so HPLC mass spec, so that improved our detection, and we were comfortable able to then measure all of the actives coming out, even in the early stages when they were relatively dilute. And then, because this was not a trial, there were a few different ways parameters that had to be investigated, had to be looked at. That the study design that we put in place, and we did it over a few different studies, was flexible design. So there, as answers were got during it, we were able to adapt and cover those in, not necessarily almost in real time. And so that meant that the project was more adaptable, and it was also more efficient. So the results that we're getting are quite good, and we're really happy with them, and so we were quite happy to work with each other. So that's my discussion then, and I maybe stop sharing. And
that's great, Stephen. Thanks very much for that. Okay. And so just hand over to Lena now, who will talk to the kind of, so it's the practical expression of that in the lab.
Hi, everyone. My name is Lena Madden. I'm going to try to share the screen now. Tim, is it visible?
Yeah, that's perfect. And if you pop that on presentation mode, that'd be super.
Yeah, I'm going to try to do it now. So, hi, everyone. My name is Lena Madden. I'm a business development scientist at Shannon ABC in Limerick. And my main specialization is analytical chemistry and these few little babies. This is our new buy, 3-bed semi in Limerick, is a triple quadrupole MS detector, which we use for very precise and very low level of quantitation. But not only that one, it has DAD detector on it that can work as a regular HPLC. This is our quadrupole time of flight, which we use for the untargeted analysis, identification, what compounds are there. FTIR, which we use for fingerprint analysis, and that could be hooked up to the gas chromatograph and do really miracles, and that one will be the only in country. As Tim showed, HPTLC and GCMS chromatogram. And these instruments we use on routinely daily basis to conduct our studies. So we use the LCMS for analysis of the sample for the profile, for the solution profile of the gel that in question that we work with SPV Healthcare on. And when the guys came to us, they had the problem, as Stephen said, with the detection and sensitivity of the method. So the elution profile was quite problematic, and we believe that was because of the level of detection. So first of all, what we did, we have improved level of detection on instruments. So it went from way below level of limit of detection to very strong to the very, very strong signal that we could comprehensively quantify everything that is coming out of the solution vessel. So, and after that, we looked at the time of dissolution profile. So that's very important that when you, when the human being take this gel, that the compounds, the APIs are released gradually over time. We looked at the pH because each individual person have their own range of pHs, and dependent on how healthy they are or how the illness is progressing, gel would have to work at different pH levels. So we looked at this the efficiency of the release at different pHs. Then we look at the compound stability, because many compounds, well, could be very valuable when kept at temperature of human body or 37 degrees Celsius. They start slowly degrading or decomposing, and then do not produce the necessary effects. So we looked at the compound stability and have shown that it all works, the profile is working, and the compounds are stable. And also we looked into the solution medium improvement to bring it as closely as possible and simulate the bodily fluids of the human body for registration and for regulatory approval when Stephen needs to apply for that. So what else how different our approach to approaches maybe of normal regular research lab. We always ensure repeatability. We will not submit our report unless we know that our results are repeatable and they're the same at every time. We study statistics on the profiles, on the dissolution profiles for Stephen, and also for all our clients, for making sure that it works every time. We have inbuilt quality control. So we're working with the European Pharmacopoeia methods. We're working with the with all the FDA and European Medical Agency directives in order to improve quality of results coming out from our lab continuously. And the test results and methods that we produce are ready to transfer. So when I'm going to be giving Stephen the results, it's not only going to be what we have achieved, it's also going to be how he can use the methods in his own laboratory. Okay, and how can he replicate? And the technology transfer is another big part of our day-to-day operation. So in every client, every company that comes to our lab, we take an individual approach. We are flexible in funding mechanisms, so we can advise, as team has explained, either to go to Enterprise Ireland for the sponsorship of the research funding, or we could advise if the company needs only very small, it has a very small question and it would be answered very quickly and easily. They could fund the projects themselves, or if they have enough capability. First of all, then we review their own processes. They will look into the process and the project and why the necessity of research have risen from the outset. Okay, and maybe what approaches we can take to solve, or maybe the company might think that the solution would be, and that happens quite often, that solution could be in one way, but we could bring our innovative solution to play and help the company to project develop and business develop or method develop for any application, for anyone and any application that comes to our door. And also we always ensure there is no client is too big or too small. We always pay the same attention and produce the same quality of the result, either from small and starting companies or for the big multinational clients that we have. And that is all from me.